Did you know that several millions of fish are used worldwide to test water samples such as effluents for its impact on fish? And that several hundred thousands of fish are used annually to assess the safety of chemicals? Under the European chemical legislation REACH, that is the registration, evaluation, authorization and restriction of industrial chemicals, fish is the most widely used test organism to assess fish acute toxicity, meaning survival, bioconcentration of chemicals, and long-term impacts on growth and survival. What I can tell you is that meanwhile we work on alternatives to using fish as experimental animals for all these three types of tests. Yet we have come the farthest for the one that is most severe, the acute fish toxicity test, where I will mainly focus on during this presentation. But also I will show you examples of the latter too. You see, probably Christine Sherman or I would have come where we are now if it was not for this amazing scientist, Niels Boles. He is a pioneer in the biology of fishes and has started many cell lines from fishes, including this one here, the rainbow trout gill cell line RTgutW1. So Christine was fortunate enough to do her PhD with Niels. And in fact, the year when Christine started her PhD at University of Waterloo was also the year of the publication for the establishment and characterization of the RTgutW1 cell line. So Christine and Niels started to ask themselves if this cell line could not be used to predict the acute toxicity of chemicals and water samples to fish, based on the assumption that in acute, so short-term exposure scenarios, it is likely most often the gill tissue that is damaged by chemicals in the water, causing the fish to die. So they established the so-called RTgutW1 cell line assay, where you seed the cells in well plates and upon attachment expose them to chemicals or water samples. One key aspect here is the use of a protein-free exposure medium that Christine formulated in her PhD based on the routinely used cell culture media Leibowitz 15, abbreviated L15. The protein-free nature of the so-called L15X allows that the chemicals or contaminants are freely available to the gill cells in the exposure media. After 24 hours of exposure, the cell viability is detected by using a combination of 3 fluorescence indicator dyes, measuring different toxicological endpoints on the same set of cells. The fluorescence intensity of this indicator dyes can be measured using a fluorescent plate reader, and by normalizing the fluorescence intensity of the exposed cells to the control cells, those response relationships can be investigated. Indeed, we kept this triple assay to this day, as it has been proven a very useful surrogate for fish survival, for fish cell viability. And what is more, since we focus on these 3 subcellular compartments, it is also possible in certain cases to deduce on specific mode of actions of chemicals on these endpoints. But it was not until 2002, at the time when Christine had already left Canada, that they were able to publish the first proof of principle to demonstrate that yes, the acute toxicity of water samples to fish can be tested with the rainbow trout gill cell line assay. So in this study, they tested 16 effluents from a paper mill, which was repeatedly sampled over the course of 1 year in a blind fashion in an acute fish toxicity test with rainbow trout and in the RTgutW1 cell line assay. And indeed, out of these 16 samples, 1 sample was toxic to rainbow trout, and this was also the only sample being identified as toxic in the RTgutW1 cell line assay. So the identical outcome in the cell assay and the fish acute test already underlined that the cell viability, in fact, can be used as surrogate for fish viability or survival. But then it was not until 2007, 2008, when Christine and I had already met, that we had a strategy ready to systematically improve this and other assays to make them applicable for environmental risk assessment. As proposed in a review by Christine on how to improve vertebrate cell cultures to establish them as substitutes for regulatory testing of chemicals and effluents, in 2006, we pursued 3 important steps. First of all, the role of physico-chemical properties of test chemicals was investigated, and the octano water partition coefficient and Henry's law constants were found to significantly explain the low absolute sensitivity of in vitro data in correlation to in vivo data. Second, a systematic selection of test chemicals for the development of alternatives to animal testing in ecotoxicology, with a particular focus on fish, was performed, coming up with a list of 60 reference chemicals spanning a wide range of different toxicities, physico-chemical properties, and modes of action. And as the third step, the dosing procedure got optimized and exposure concentrations controlled by implementation of chemical analysis of the exposure concentration. So after all this was figured out, it were Katrin Tanneberger and I, with the support of the CEIISens team, who tested about 35 chemicals of this systematically selected list of 60 reference chemicals for their toxicity in the gill saline assay, and compared quantitative effect values to those available for fish. And what you can see is a very good quantitative correlation that we observed. The majority of the test results falls within a 10 or even 5-fold range of the line of unity. But as exceptions prove the rules, we also found some outliers. So most notably here are permethrin, lindane, and caffeine, which are neurotoxic chemicals that act through specific channels that cannot be well predicted with the simple nature of the gill saline assay. So for the time being, this group of chemicals needs to be viewed with caution for this application. Overall, the results looked so promising that we witnessed great interest by research and from academics, but also from industry and from regulators, so much that we received funding and also motivation to perform a round-robin study to assess the robustness and the Intra and Inter-lab reproducibility of our test. So at first, we again selected a set of chemicals with different physico-chemical properties, mode of actions, and toxicities for being tested within the round-robin study. Then I trained our test partners at the Eawag labs, who then transferred the test to their lab. And also during this training and transfer process, some simplifications and adaptations on the protocol were made. Just briefly again on the test procedure. The cells are seeded to 24 well plates and afterwards are exposed to a dilution series of test chemicals for 24 hours with following determination of cell viability using these 3 fluorescent indicator dyes. What I want to highlight here is the sampling for chemical analysis that was performed at the start and the end of the exposure out of the wells. And by doing so, we were able to base the evaluation of test results on the geometric mean of the measured exposure concentrations at start and end of the test. The participants tested each chemical in at least 3 independent runs and provided me with the raw data of their fluorescent readout. What we were able to do was to verify that the assay is robust. All partners were able to establish the cell line and the assay in their labs, even though none of them had worked with these cells before. And all were able to perform the assay according to the quality standards set. Here you see the example of one chemical and how the results looked in the different labs and between the labs overall. And we found values for intra and interlaboratory variability that are in fact very well comparable to bioassays with similar formats. Specifically for 2, an acute toxicity test using zebrafish embryos, which is an OECD guideline since 2013, and a fish hepatocyte assay, which is used to predict bioconcentration factors in fish and which was adopted by OECD in the middle of last year. So with all these data evolving, we increasingly found support to apply for international standardization. And at this point, I would like to express great thanks to Adam Lillicrap, a colleague from NIVA, an environmental science institute in Oslo, who contributes to the section in ISO that deals with water quality. He invited us to send a proposal to ISO already back in 2014. And after all rounds of commenting, voting, and further improvements of the method descriptions, we are very proud that our assay was accepted in April last year as ISO standard 21115. So is that the end of the journey for the RTgutW1 cell line assay? Well, no, after all, it's the journey that matters anyway, isn't it? So first, since the assay has reached international acceptance, we wanted to make our expertise and know-how available to everyone in need to screen chemicals or water samples for acute fish toxicity. And on this background, we have started an Eawag spin-off, Aquatox Solutions, that offers this test along with other animal-free tests for environmental risk assessment. Moreover, we have submitted a proposal to OECD to evaluate our proposal as test guideline on this level as well. Our proposal has been accepted and is now part of the OECD test guidelines program. And getting the protocol accepted by OECD would mean even a wider use, especially for chemical testing. And scientifically, yeah, there are more challenges ahead. For example, while the assay itself is completely animal-free, so there are also no animal components such as serum used, the routine cultivation of the cells still requires some serum addition. As it stands now, with the serum of one calf, we can currently carry out the cell line assay to save 6,400 fish. But clearly our aim is also to eliminate this last animal-derived component. The happier we are that we could secure funding through the first open call of the Swiss 3RCC from last year to carry out a research project to better understand the nutritional requirements of fish cell lines and to develop a medium that allows fish cell line proliferation serum-free. So now to the other examples. Another approach where we work with the RTgutW1 cell line is the prediction of fish growth based on in vitro data, a project led by my colleague Julita Staniczka-Michalak. So the basic assumption here is that reduced fish growth after chemical exposure is correlating with reduced cell survival and proliferation. So a fish that grows less has fewer cells in the body, and this is what we can measure in the culture dish. So by counting the cell number and converting it to cell weight, we can translate the reduced cell weight by computational modeling. To test this hypothesis, we predicted the impact of cyproconazole and propiconazole on the growth of rainbow trout and fathead minnow, respectively. So each black dot you see in these graphs represent one fish, and the blue and the green lines are the respective average values. And now the red squares represent our model predictions based on cell line data from 5 days of exposure in the lab, whereas fish were exposed for 31 or even 62 days, depending on the species. In my last example for rainbow trout fish cell lines as alternatives to fish in risk assessment, a set of 3 cell lines got used in a case study for the prediction of bioconcentration factor of benzoate-pyrene. The used cell lines were the rainbow trout W gill cell line that you already know, as well as the rainbow trout liver cell line, RTL-W1, and the RTgutGC cell line from rainbow trout intestine. They got selected for the assumption that these 3 organs are the major uptake and biotransformation site of chemicals in fish. So by monitoring the chemical depletion over time In these 3 cell lines, in vitro biotransformation rate constants can be obtained and incorporated into a physiologically based toxicokinetic model. The multi-compartment PBTK model accounts for chemical uptake, internal distribution, and elimination from rainbow trout, as well as for its biotransformation in different fish organs, as is determined in vitro in the different cell lines. The application of the cell line derived in vitro clearance rates into this PBTK model predicted a bioconcentration factor for benzoapyrene of 909 to 1057 in the approach where all 3 cell lines were combined, compared to 920 reported for rainbow trout in vivo. Although the liver cell line RTL-W1 was most efficient in clearance of benzoapyrene, the consideration of all 3 were found to more fully account for whole body biotransformation in vivo, resulting in a highly comparable bioconcentration factor value. For both of these last examples that I showed you, the biotransformation and the growth, we've already tested several more chemicals via Cefic LRI and Swiss 3R project fundings, and could confirm these proof of concept studies. With that, I'm at the end of my story on how to use fish cell lines of rainbow trout as alternatives to fish and environmental risk assessment. And I want to say a big thanks to Eawag for its supportive environment and giving us the possibility to go for such standardization processes like ISO and OECD. Christine for all her positive energy and for being a great friend and role model to me, not only scientifically, and the 3RCC who acknowledged our work on the gill cell line assay by conferring last year's Swiss 3RCC award to Christine and me. And of course, thank you for listening to this webinar.